This is a podcast of the Nuffield Department of Medicine. Today we're speaking to Dr. Benjamin Schuster-Böckler about his research in cancer informatics. Hi, Benjamin.
Hi.
What is bioinformatics?
Bioinformatics is the field of research that tries to answer biological questions using computers or computational tools. And it's really a fairly new field. Over the last 20 years or so, there's been a revolution almost in the biological sciences with new experimental techniques that produce huge amounts of data. When I say huge, I mean for one cancer patient, you would produce so much data that it would take you probably a whole day to download it on your home internet. But we don't have one of them. We have maybe thousands of those data sets. And really, it's almost impossible now to do research without using computer programs to actually sift through that data to find the biologically relevant aspects and distinguish them from random noise.
What are the most important lines of research that have developed in the last 5 to 10 years?
I found particularly striking what has come out of the genomics revolution in the last 5 or 10 years is that we're now aware that cancer is an incredibly diverse disease. It's almost as if it's not just one disease, but it's many diseases. And each patient can have very similar presentation in the clinic, but if you look at the molecular details and their genomes, these patients can be incredibly different. And it's really becoming more and more clear that those differences can have a great impact on the treatment success of these patients. So this field of cancer heterogeneity, as it's called, is one of the things that I found particularly exciting.
How is bioinformatics helping us understand cancer?
Nowadays, with the explosion of data that's being generated in cancer research, it is almost not a question of how it can help us understand cancer, but it is almost impossible to do any cancer research anymore without the help of computer tools. To sort of put this into perspective, it's very common nowadays to generate data sets which have millions of rows and tens of thousands of columns. And by just looking at them, you would not be able to understand what they mean. So we really need computer programs to help us detect the biologically relevant aspects of the result and distinguish them from those aspects which are noise, effectively.
Can you tell us about your research?
So the research in my group is really focused on trying to understand the origins of variability in cancer. And maybe to sort of explain that a bit better, I can use an analogy. So a mutation in the genome is a bit like a car accident that happens on a stretch of road. But not all roads are equally dangerous. So even though each accident might be bad luck, the frequency of accidents varies quite massively between different roads. And really what we're trying to understand is what is the overall frequency, what's the sort of the specific likelihood of an accident in a particular area on average. But then the important question is, are there specific roads where the accidents happen with much greater frequency than on average? And those ones are often the regions that are of particular importance for disease. So in the genomic context, there are some regions in the genome which accumulate mutations at much greater rate than similar other regions in the genome which have similar characteristics. And those ones are the ones that we're trying to identify.
Why does this research matter, and why should we put money into it?
We can see now that patients with cancer are very, very different on the genomic level. And it is crucial for us to understand which ones of the mutations in the patient genomes are really driving the cancer, which mutations are the ones that are really important for the development and the progression of the disease, and distinguish them from those variations which are just, as we call them, passengers. And in order to be able to distinguish those drivers from the passengers, we need to have an idea of what the overall likelihood of mutations is in the genome on average, because that will allow us to really pinpoint those mutations which happen at much greater frequency. And those ones are usually the ones which really are relevant in the disease context.
How does your research fit into translational medicine within the department?
I think it's generally accepted that in the future we'll see a lot more personalized medicine. When I say personalized medicine, I mean that we will start tailoring treatment to an individual patient's genomic characteristics. But in order to be able to do that, we need to understand which aspects of an individual's genome are really predictive for treatment success and which ones have absolutely no impact on the disease. And so one of the key aspects of the research that we're doing in our lab is helping to make that distinction.
Thank you.
Thank you.